Hi, everyone. So these are the answers for the genetics review. We ran out of time in class, so I wanted to make sure that you had access to some explanations for all these crosses, especially because most of them are 2 gene crosses before the test. And I believe one class actually finished this problem that I'm looking at right here, but I'm going to go ahead and start here because the other class didn't quite get through that, okay? So keep in mind when we're doing this, you need to have your gene tracker and all of the necessary papers that tell you what genes are on what chromosomes and what the alleles are. So make sure that you have that handy when you're going through this, just in case you want to try them on your own. Okay, so I got my gene trackers, I got my karyotypes, so let's go. So this is the female side again, that's her genotype. This is the male side. So when setting up allele combinations in the eggs, this is the information that we'll use. Keep in mind that this is one allele for the head spike gene. This is the other allele. And for a second gene, this is the skin color gene on chromosome 3. That's one allele. And that's one allele. So she's heterozygous for both of the genes that we're considering. So again, if you want to be really careful here, you can do the foiling to make sure that you get all of the allele combinations and that you can consider independent assortment. So what I get from this is, for this one right here, capital H, capital S with a capital B. But capital H, capital S can also pair with that recessive B, okay? We also get 3 and 4 here, so those combinations will be this with this, and this with this. Luckily for the male, it's the same genotype, so it'll be the same allele combinations except in sperm. So I can just go ahead and write those out across the top here in a similar manner. And then I'm going to go ahead and make my giant 16 squared Punnett square. And if you recall from class discussions, if both of these genes behaved in a predictable Mendelian fashion, that is, if one of the alleles was dominant, clearly dominant, and the other allele was clearly recessive, I could go ahead and say 9 to 3 to 3 to 1 and know that that is the answer. 9 being double dominant, 3 being one dominant and one recessive, the second 3 being the other dominant and other recessive, and the 1 being the double recessive. But if you're looking at the gene tracker, we know that the skin color gene has alleles that behave in an incompletely dominant manner. So while the percentages overall of how these alleles behave won't change, what we have to account for in the actual Punnett square is the representation, the numbers. Okay, so I'm going to go ahead and quickly add these. Hopefully my speed doesn't lead to any incorrect genetics, but keep an eye on me, make sure I'm doing this right. Of course, the real time-consuming part of this is you have to go back and interpret all the genotypes from the crosses in a moment here. And once this is actually done, you could just memorize what it looks like when one gene behaves in an incompletely dominant manner and when one doesn't. But let's go ahead and be careful and actually do the cross. Okay, so then we have to go through and interpret what all this is. So let's clear some space for that. I'm going to put these over here. Just to make space. So I guess that's problematic in that you won't be able to double check to make sure that your alleles are correct, but we'll figure that out in a minute here. Okay, so here's how I go ahead and do this. You can go ahead and just circle the one that you're working on and cross it out when you're done. So if I'm looking at this organism, I have my gene tracker right here. I'm seeing that it's going to have a sharp head spike and it's going to be blue. So I'm going to say sharp, blue, and I'll just keep a tally. Okay? This one, sharp, but green. Right? This one is sharp green because it's incompletely dominant in your heterozygous there. This one is sharp blue. This one is sharp green because of the heterozygous. This one is sharp green. This one is sharp. And this is our first instance of a yellow, so sharp yellow. Sharp green. Sharp yellow. Sharp blue. Sharp green. This is our first instance of dull. So we have dull blue. Dull blue. Okay. Another dull, but it's green. Sharp green. Sharp yellow. Dull green. And our first and only dull yellow. Okay, so we only have one of those. Now here's the interesting thing that you can go back and do. So we have our combinations here. You can see that it's not the predictable 9-3 to 3-1 that you see in the classic dihybrid cross where both of the genes are completely dominant. But there is some predictability into it. So if you consider this head spike gene because it's traditionally dominant, you can see that sharp, which is the dominant, and dull, which is the recessive, should be about 75% to 25%. Obviously, the numbers can skew a little bit, but if you're looking at a large collection of progeny, you should see 75% sharp and 25% dull. So if that is out of 16, in other words, you should see 4 dull and 12 sharp. So if I'm counting these up, the sharp, 3, 6, 6, that's 12. So these account for those 12 sharp. And then if I'm looking at the dull, 1, 2, 3, 4. So there's your dull. So that works out. The math works out there. Likewise, even though incompletely dominant, you should still see the traditional 25, 50, 25 in a basic skin color incompletely dominant cross like we've seen with petunias, flowers, and other examples of incomplete dominance. So that would be 25% or 4 blue. So your 4 blue would be the 3 here, the 1 here. You should also see for yellow, that's the homozygous recessive. Here's your 1 here and your 3 here. And you should see 50% heterozygotes showing the intermediate incomplete dominant phenotype. In this case, that's green. So here you have 2 plus 6. So 6, 7, 8. Yep, that's the 8. That's the half. Okay? So, you know, you could memorize this. I think memorizing the Mendelian stuff is probably better, and then not memorizing the non-Mendelian stuff and just doing the crosses for those non-Mendelian examples is probably a better idea. You can see it's a lot of work, but you can get there if you just kind of stay focused and make sure you keep track of all of your alleles. Okay? Okay, so let's go on to the next example. Here we're looking at the skin color gene, the same incompletely dominant skin color gene, which obviously requires a lot of work, and the antenna gene, which if you're looking at your gene tracker, you'll recognize that the gene for antennae is codominant and has multiple alleles. Okay, so we're going to have to keep track of that. Again, it doesn't complicate the genetics at all. You're still going to make a Punnett square. You're still going to assign gametes with certain alleles and making sure that you have all those allele combinations. The problem is interpreting the genotypes and making sure you account for the proper phenotypes when counting your progeny. Okay, so in this case, you have to understand what incomplete dominance is, what codominance is. Okay, so let's go ahead and create some gametes. Okay, so again, you could foil if you like. This ensures that you get all the combinations. So again, my number 1 would be capital B, ooh, that's tricky. Capital B, lowercase c, lowercase b, capital B, and then lowercase b, lowercase c. So here is an interesting thing. Genetics is abstract. We're assigning symbolic representations for what our alleles mean. So we've sort of run into a problem where we've assigned similar symbols for different genes. So we'll just have to be careful of that when we take that into consideration. Now, you can't just make the same allele combinations for the gametes, because while this other organism, while the male is in fact heterozygous as well, he's heterozygous for one of the different alleles under the antenna gene. Okay, so we're just going to be very careful and foil this out again. Okay, so with this, I get a capital B, capital A. I also get a capital B, lowercase c, lowercase b, capital A, lowercase b, lowercase c. Okay. Let's go ahead and draw up our boxes. Again, we're just being on the safe side here. So when I'm looking at this, I see the following. Okay, so those are all the genotypes of possible progeny. You know, if you're looking at 16 individuals or 32 or 64, however many you're looking at, you should see this predictable percentage of genotypes and consequently the same related percentage of phenotypes. So let's go ahead and clear a space here for our interpretation of the progeny. Okay, ideally if you were working on a piece of paper, you wouldn't erase everything, you just make another space, but I'm out of space on this screen. So again, we're just going to go through and look to see. So if I'm correct looking at this individual is blue and has circle and square antennae. Okay, so I'm going to say blue and both. Right, we're saying both because for this organism, in this particular, set of genetics, when you have a capital B and a capital A allele for antenna, you have both. Okay, so this organism would have both. Okay, this organism would also be blue, but we can't put it under the previous header because in this case they just have A antenna, which equals circles. Okay. So circle antenna. Okay. Now we have our first green individual. Remember this heterozygote is green. So they would be green with both. Green A. Green A, which is circles. Blue B. B, which is squares. Blue none, right? This would be a blue none because the homozygous recessive alleles for antenna doesn't create anything there. So blue none. Make sure I'm keeping track of all these. Okay, we have a green B. B is squares. Okay. We have green none. So this would be a green none. And we got lots of different types of progeny here. Okay, so we're going to run into a green both. We're going to run into a green A, which is circles. This is our first yellow. So we have a yellow child that is both. Okay, we have a yellow circle right here. We have a green B. Green B is square. Okay. We have a green none. We have yellow B. Yellow B would be square. Okay, and we have a yellow none, which I'm running out of space. Yellow none. So I think we may have gotten every single possible phenotype there. For the yellows, there's only one of each of the possible combinations for yellow. But then you see we have green, we have the blue. So, you'd count it up. That's sort of a nightmare. So I would take an example like this, I would take as fractions. So, for example, if we're looking at this one, you have one that is blue in both. You have one that is blue in circles. You have one that is blue in square. You have one out of 16 that is blue in none. So I'll make sure that I put the fairest instructions in there. Okay, moving right along. This is getting kind of long. So we're going to look at the I number gene. Okay, so I number gene. is shown here. And we're also going to look at we're going back to the head shape gene. Okay, so let's do that. This with this, this with this. This makes it nice and easy, right? So because for the I number gene, the male over here is homozygous recessive, so that eliminates half of the work, right? So we just have this possible combination and this possible combination. Guys, do not do this. Don't repeat, right? You've already done that one here, you've done that one here, so that's just extra work. Don't do that. Okay, so let's interpret what our progeny will look like. Careful there. You can see me flying through these crosses. Hopefully you understand, and that's underlined, that it's pretty easy to make a mistake if you go too fast here. So ideally when you're doing this, slow it down, take your time. There won't be a whole lot of questions on this test, so you'll have plenty of time to consider everything that you're looking at. So okay, let's account for what our progeny look like. So according to my gene tracker, anything with a capital E, because it's clearly Mendelian, clearly completely dominant, will have two I's, okay? So I'm looking at this right here. Anything with a capital S will be square. So this is two I's, square head, two I's, square head, two I's, square head, two I's, ooh, two I's. That's homozygous recessive, so that would be two I's and a round head. Okay, now on the bottom here, this is where we have our individuals that have one eye, because in this case, they're homozygous recessive for that I gene. So one eye square, right? One eye square, one eye square, one eye square, and then you're going to have a group that's one eye and round. Okay? And that's just this one right here. Okay? So you could, easily convert that into 3 out of 8, 1 out of 8, 3 out of 8, 1 out of 8. Okay. Eye number and eye color. Okay? So the only reason this is tricky is because the eye color gene is sex-linked. So it's on the X chromosome. So you have to keep track of, males and females that have each of the phenotypes. So that's the cross. And again, homozygous recessive for the I number gene, so that's kind of nice. So I like the way this is organized because over here basically you have the male data. Over here you have the female data as far as sex goes in these crosses. So just be aware of whether I'm asking for a total population representation of phenotypes or if it's male versus female. So let's represent the whole population. This individual here, again, if you have a single capital E, dominant E, you have two eyes. So this would be a two-eyed female, and because capital R is dominant over the lowercase r, you'd have red eyes versus white eyes. Okay, so this would be a, and we're not going to keep track of the sex, but I will tell you as we're doing it what they are. So for population, this would be two eyes, red. Again, we have a two-eyed female, but in this case she's white-eyed. Two eyes. Only because she has the lowercase r. Okay, another female here. One eye, red. One eye white. Okay. So for the males over here, this individual here would have two eyes and be red, and a male. This one would have two eyes but be white. This one here would have one eye and be red. And this one here would have one eye and be white. So population-wise, it's 1 to 1 to 1 to 1, right? 2 out of 8, 2 out of 8, 2 out of 8. Okay. The last one I want to cover is this example of stripe genes. Okay, so this is an example of polygenic trait. Okay, so we're going to cover polygenic traits in this one. That is to say, both of these genes, right? So this gene and this gene contribute to stripedness, to the number of stripes in an organism. Okay, so when we're looking at this, one of the alleles for each, the capital one, is dominant and the other one is recessive. Okay, so what's neat here is that on this side over here, for the female, if you were looking at the correct karyotype, just making sure that I am. So she's homozygous recessive here and heterozygous here. So what's nice about that is you only have two combinations. Everybody gets lowercase st. Half of them will have a capital F. Half of them will have a lowercase f. Neat thing about the male is, again, homozygous dominant. So everybody will have a capital S, capital T. Half will have a capital F. The other half will have a recessive. Okay? So here's what I look at this as. So I'll go ahead and do the genetics, the cross. Okay, so how I kind of keep track of this, because there are two genes that are involved in the same phenotype, I kind of make 4 little bubbles that represent, for this individual, the number of dominant versus recessive alleles. So, you know, right here, that's dominant. So I'm going to bubble that in. The second one is not dominant, so I don't bubble it in. But the third and fourth, this individual is homozygous dominant for that second stripe gene. So I just kind of bubble them in to get an idea of the number of dominant versus recessive alleles that these individuals have. Okay, and then I sort of keep track of, well, individuals that have 3 dominant, 1. Individuals that have 2 dominants, it's 2. Individuals that have 1 dominant allele is 1. So if I'm looking at stripes, you have 1 individual that has many, you have 2 individuals that have some, and you have 1 individual that has few, right? Notice that we don't have any individuals that have all dominant stripe alleles. We don't have any individuals that have no stripe alleles. So the offspring will all have stripes to some varying degree. One of the offspring, 25% roughly, if you were to have 100 kids, maybe 25 have few stripes. Vast majority would have some. And then you'd have 25% that would have many, that have lots of stripes. So this is an example of a polygenic trait. A lot of human traits are polygenic in nature. The classic example is skin color. There are many genes that contribute to that. And just generally speaking, the more dominant alleles you have associated with pigmentation, the darker your skin is, right? What gets really interesting is if you do an F2 cross, that is, if you take these progeny here, and you perform another cross to see what happens, in the next generation. So if you wanted to, you could try that, but we're just sort of making sure that we get this basic polygenic idea down. Okay, that's become very long. Thanks for tuning in and doing your due diligence for this genetics test coming up. Okay, talk to you soon.